Good afternoon, students. Good afternoon, students.
Good afternoon, sir.
Good afternoon, sir.
Okay. Okay, so students, we have been talking about antibodies, right? So we have already talked about IgG and IgM, right? So, uh, who wants to tell? So just raise your hand. How many subclasses of IgG are present? Students, yes, Manaswini?
So 4.
Okay. Okay, so my next question is, which IgG subclass is most effective complement activator? So just raise your hands. Which IgG subclass is best in activating? Yeah, Sanjana?
The IgG3.
Yes, IgG3, it's the best activator of the complement system. Okay. The pentameric form of immunoglobulin is seen in which class? The pentameric form, yes, Shakti?
IgM.
Okay, good. IgM. And what is, okay, next question is, what is the role of J chain? Yes, Yuvraj.
is used to join.
दोबारा बताना.
So it is used to join.
Yes, it helps in joining of the immunoglobulin molecules and helps in the formation of disulfide bonds. Okay. Okay, so now who wants to tell about the secretory component? Anyone? What is secretory component in IgA? Anyone? Okay, so let's start with IgA. Okay, so I think I started and we just uh, discussed one slide of IgA in the last class. So let's continue from there. So students, do you have any question or query from any of the previous class? Any question, query from the previous class from IgG, IgM, or any question even earlier to that? No?
No, sir.
Okay. Okay, so we'll start with Okay, so I think we have already started with this slide. So let me just refresh this. So this is immunoglobulin A, right? And 10 to 15% of the total serum immunoglobulin. So you'll find immunoglobulin A. It's the most important player in mucosal immunity, okay? And immunoglobulin A is the one which is present in the external secretions, right? So you will find immunoglobulin A in milk, in saliva, in tears, then it is present in the mucus of bronchial, genitourinary and digestive tracts, right? So you can see its position. It's there in the milk, it's there in the saliva, in tears, right? And all the mucus that is there in our body. So immunoglobulin A will target uh, the pathogens in the mucus region, right? Or in the eyes. So the tears, they contain IgA, right? And they have some other enzymes. So when the pathogen gets into your eyes, so it is trapped by IgA. Okay. So this again, I think we just saw in the previous class. So when this immunoglobulin A is present in the secretions, it is in a dimeric or tetramer form, right? So dimer is the most common form of IgA that you see, and it includes two molecules of IgA. So you can see here one molecule, two molecule, okay, and it has a J chain. So the J chain helps in joining of the two molecules and helps in the formation of disulfide bond, okay, and it also has a secretory component, okay. So we will talk about secretory component as well. So secretory component uh, protects this immunoglobulin A, okay, from being degraded by the proteolytic enzymes, okay. So you can just imagine एक dimer है which is joined by some disulfide bonds, right? And if this dimers get into some environment where there is a high amount of acid or various degradative enzymes, so it can break the bond. Okay, so secretory component, it protects. S is secreted, okay, or the secretory immunoglobulin A protects IgA from being degraded, okay, by proteolytic enzymes. So this secretory component which is around here, it helps in providing protection to the IgA dimer, okay, and that is the reason why this IgA dimer can survive in the harsh gastrointestinal tract environment, okay. So because of the secretory component, this IgA dimer is able to survive in the harsh gastrointestinal tract environment and provide protection against microbes that multiply in the body secretions, right? So you can understand the importance of this secretory component, okay. Now in the next slides, I will also discuss from where this secretory component is coming, okay. And IgA is a poor activator of complement system and opsonizes weakly. Okay, so it is not good in activating complement system or opsonization, right? As what we have seen in case of IgG and IgM. Okay. Now let's see the process of IgA dimerization. Okay, so we have already seen pentamerization of IgM. Okay, so that was a complicated step where five molecules were getting together. Okay, but when we are talking about a dimer, only two molecules have to come together. Okay, so exist in serum as monomer. We have already talked about that, but more usually as a J-linked dimer, okay, in okay. So the process of dimerization is similar to that as IgM pentamer, right? So we have already seen this. So you just need to imagine this is your Fc region, and then you have the Y arms here. Okay. Now what happens again? Which molecule will help? It will be the J chain. Okay. So the J chain will come, and then it will lead to formation of disulfide bond. Okay. So this J chain helped in the formation of disulfide bond and now the two molecules have come together. They are joined by this disulfide bond. Okay. Now when we saw the pentamer, what happened? The J chain, it got detached and it bound with the next molecule, right? But in this case, you don't have any third molecule coming here, right? So the J chain does it work and it stays there. Okay, so this is the process of IgA dimerization. So as compared to IgM pentamer, it's very simple, right? So IgA now exists in two subclasses. Okay, you have already seen that IgA is present in two subclasses. One is IgA1, which is mostly found in the serum, okay, and made by bone marrow B cells, whereas IgA2, this is the one which is mostly found in the mucosal secretions, colostrum and milk, okay, and is made by the B cells located in the mucosae, right? So you can see IgA1, it is there in the serum, okay, and where it is made, it's made in the bone marrow, right? Whereas IgA2, it is found in the external secretions, okay, and it is made by the B cells which are present near the mucosa, okay. So this is the major difference between IgA1 and IgA2. Now let's see the difference between IgA1 and IgA2. So two isotypes, IgA1 is almost 90%. So if you see 90% of immunoglobulin A is IgA1, okay, and 10% of immunoglobulin A is IgA2, okay. So IgA1 is in higher numbers as compared to IgA2, okay. IgA1 is the predominant immunoglobulin A subclass found in serum, right? So we just saw this in the previous slide also. IgA1 is more in serum, okay, and IgA2, uh, it is in the external secretions, okay. So that is the major difference between IgA1 and IgA2, okay. Now let's see what happens when there is IgA deficiency. Okay, so decrease or absent IgA, which is termed as IgA deficiency, can be a clinical significant immunodeficiency. So uh, there are some people, right? So they have some genetic defect, okay, and those people, they are not able to make IgA in their bodies. Okay, uh, so obviously, when there is deficiency of IgA, or for that matter, any immunoglobulin class, the person becomes susceptible to various infections. Okay, I'm not going to discuss that here because we are going to talk about primary immunodeficiencies later. Okay, maybe it's in your syllabus. So sometimes what happens, some people, they have some genetic defect, and because of which they're not able to produce some components of the immune system, okay, For example, a person may not be producing immunoglobulin A, okay, and so therefore whatever protection a person gets through these external secretions or through the mucus, so that protection is lost, okay. Now, Neisseria gonorrhea, I'm sure you know it causes a sexually transmitted disease which is gonorrhea. Okay, it releases a protease which destroys immunoglobulin A. Okay, so if you remember, I said that IgA is present in the genitourinary tract. Okay, so uh, this pathogen, Neisseria gonorrhea, it will, so it is a sexually transmitted disease, right? So IgA is there to fight Neisseria gonorrhea, right? But Neisseria gonorrhea is also smart, okay? It has also evolved over a period of time. It knows that we have IgA, so it has also developed its own methods to tackle this, right? Because see, even the bacteria wants to survive, okay, and they also evolve, they also develop, they also see how they are being killed, okay, and they develop their own strategies. Just for example, what we see in antibiotic resistance, right? So those pathogens, uh, they sense the environment, okay, and change themselves so that they can survive in that environment, okay. So Neisseria gonorrhea, very interesting. It releases a protease which destroys IgA. Okay, so IgA is not there then. Okay, and it is safe, and then it establishes itself. Okay. Now IgA nephropathy is caused by IgA deposits in the kidney. So sometimes what happens? IgA, immunoglobulin A, it gets deposited in the kidney, okay, and because of that uh, it can lead to some disease condition, okay, it can cause damage to the kidneys, okay. So again, I will not talk about this nephropathy in detail here because again, when we talk about uh, hypersensitivity, right? So in one of the cases, these immune complexes they get deposited. Okay, तो जब हम hypersensitivity की बात करेंगे तब हम glomerulonephritis और nephropathy की बात करेंगे. Okay, so this is just to make you understand that uh, how IgA is important, right? And it can also lead to various disease conditions. Okay. Now this process is very important. Okay. See, अभी तक हमने जितनी बात की म्यूकस म्यूकस में secretions, IgA is there in the external secretions, right? But the question is how it comes into the external secretions. Antibodies are made inside our body, right? So they are being produced in the blood. ठीक है, तो फिर वो ब्लड से बाहर कैसे आ गई, right? How they are coming into external secretions, how they are coming into the mucus. Okay, so it happens by a process called transcytosis, okay, which is basically the immunoglobulin A antibody transport across the cell. So this process is called transcytosis. Okay, so I'm already showing the picture here. I will show you an animation of this and I will show you also a video of this. Okay, so this process of transcytosis will be clear to you, right? Okay, and this is a very simple thing. So you can just see a lining of epithelial cells, right? So this is the lining of epithelial cells, right? Now this is the submucosa region where you have the B cells which get differentiated into the plasma cells, right? And what is the role of plasma cell? The role of plasma cell is to secrete antibodies, right? So this plasma cell, it is producing IgA, okay, And once this IgA comes out, it becomes a dimer. Okay, so you know the process of dimerization we have already seen. The J chain, it joins the two molecules of IgA. Okay, now what happens? This dimer, it goes and bind to a receptor on the epithelial cell. So this receptor is called the poly Ig receptor. Okay, and students, you can see it has 5 domains, 1, 2, 3, 4, 5, right? So it is, the domains are almost similar to that of what you see in antibodies, right? So that's why it is called polyimmunoglobulin receptor. So this is the point where immunoglobulin binds, right? So you can see the IgA dimer, it has bound to this receptor, right? Clear? Now, when it has bound this whole complex, right? So, when this binding takes place, this whole complex is then taken up inside a vesicle. Okay. So now you can see the this IgA dimer and the poly Ig receptor. It has gone inside a vesicle, right? And this vesicle contains a lot of degradative enzymes, because of which this is cleaved. Okay, and where it is cleaved? At the base of the stalk, right? So now you can see it is cleaved here at the base of the stalk of poly Ig receptor. So this uh, IgA dimer then moves out. It has a J chain, and now it has this secretory component. Okay, so students, is it clear from where the secretory component is coming? So this secretory component is basically part of the poly Ig receptor. Okay, so it during this transport, it just binds to the IgA dimer. Okay, and it then provide protection to this dimer in the harsh conditions. Okay, so is this picture clear? I think it's a very simple thing. Is it clear?
Yes, sir.
Okay, so let me show you this in animated form, right? So this is your epithelial cell. Clear? Now what we are saying is the B cells located in the submucosa, they produce the dimeric IgA. So you can see it's a dimer of IgA. Again, I'm saying this is not the full picture of uh, IgA, because it will also have this Y chains. Okay. So now what happens? Okay, now this dimer, it goes and binds to the poly Ig receptor, so polymeric Ig receptor. Okay, so where they are expressed on the basolateral surface of epithelial cells. Okay, now these poly Ig receptors will capture IgA, which is produced in the mucosa. Okay, so now the B cells, they are producing the dimers. IgA are binding to these dimers. So then what happens? The polyimmunoglobulin receptor and IgA are internalized in a vesicle, right? So you can see this whole complex has now been internalized into a vesicle. And what happens in the vesicle? Because of various degradative enzymes, the stalk um, of the poly IgA receptor, it is cut. Okay, so it moves out and it now cuts. Okay, so the stalk of the poly IgA receptor is degraded to release IgA containing part of the receptor, right? So you can see here there is a part of this receptor which is called the secretory component, right? And now it has moved into the external secretion, right? So what it is containing? It is containing two molecules of IgA. It is having the J chain and the secretory component, which is coming from the poly Ig receptor. Okay, so students, clear? Any questions? Okay, if there is no more question, then I can show you the video of this process. Okay.
Is a type of transcellular transport by which various macromolecules, such as several enzymes, proteins, and antibodies, are transported across the interior of the cell. So, one side, the proteins come in, get endocytosed, and from the other side, it gets exocytosed and released. This kind of mode of transcellular transport is important for several secretory molecules. And we will take an example to understand this process. One of the most important antibodies that are secreted in breast milk, tears, or saliva is IgA. We would look at how IgA antibody is transcytosed. So we look at the breast inside which there are mammary gland. Now here is a small zoomed in version of the mammary gland. And if we zoom in further, we would see the epithelial cells and the lumen where we can see even the milk droplets. Now from the outside of this epithelial surface, the antibodies has to go inside into the lumen from where it would be secreted along with the breast milk. So in a bigger picture, we can understand outside of the epithelial cell surfaces, there are antibody producing plasma cell. So it produces IgA type antibodies, which then bind to poly IgR receptor on the epithelial cell surface. Then it gets endocytosed from one surface. Second, it would transported along the transport route of the cell and get exocytosed from the other surface. It has been seen that disrupting the Golgi architecture by brafildin A can disrupt this transcytosis. That means this transcellular transport is Golgi dependent. So this is an important process because this is how several secretory antibodies are secreted in the bodily secretion like breast milk or saliva, etc. I hope you enjoyed this video. Apart from uh,
So students, is it clear? Any questions? So students, any question, any query?
No, sir.
No, sir.
Okay, is it clear to everyone?
Yes, sir.
Okay. Okay, I think now we can have your attendance, and after attendance we can start with some uh, new class of antibody. Okay, so students, I'm going to start the attendance. Yuvraj,
Present, sir.
Yuvraj, okay, Yukta,
Present, sir.
okay, Vivek, Vivek, Tridiv, Tridiv, you are not visible.
So am I visible now?
Yeah, Tanvi,
Present, sir.
Shivani,
Present, sir. Present, sir.
Shakti,
Present, sir.
Shakti, you were not visible.
Am I visible?
Okay, Sanjana,
Present, sir.
Sahiba,
Present, sir.
okay, Rupal,
Present, sir.
Nehal, Nandini, okay, Namrata,
Present, sir.
Manaswani,
Present, sir.
Kshitij,
Present, sir.
Janvi,
Present, sir.
Ishita,
Present, sir.
Devanshi, Devanshi, Sam,
Present, sir. Present, sir.
Ayushi,
Present, sir.
Etoeba,
Present, sir.
Adish,
Present, sir.
okay.
Sir, I'm
Yes,
also
Vivek.
present.
Okay, okay, fine. Okay, students. So you can ask whatever you want to ask. Okay, students, then we'll just summarize IgA with the help of a video. Okay, the video shows the different things that we have already discussed. Okay, so let's see this video, and after the video, if you have any questions related to IgA, we can have a discussion on that. Okay, and then we'll end the class, and after that, we have another class in which we will talk about IgE and IgD. Okay.
Will study the structure, properties, and function of immunoglobulin A. Besides this, we will also understand the difference between secreted and secretory immunoglobulins. Immunoglobulin A is found in blood, but it is predominant in the secretions of the body. For example, in the mucus, mother's milk, tears and saliva. Structurally, IgA consists of 2 light chains and 2 alpha heavy chains. Each heavy chain contains 1 variable domain and 3 constant domains. There are 2 subclasses of IgA, designated as IgA1 and IgA2. IgA1 is mostly found in the serum, whereas IgA2 is predominant in the secretions. These 2 subclasses differ from each other in the hinge region. As you can see here, the hinge region of IgA1 is almost double of IgA2. Each subclass of IgA can exist in 2 main forms in the body. These are secreted IgA and secretory IgA. Let's understand these 2 forms. Secreted immunoglobulin A. It is written as sIgA here. Note that alphabet S is in lowercase. It is also known as serum IgA since it circulates in blood. It can be a monomer, dimer or rarely trimer. Polymeric form of IgA is formed when 2 IgA monomers are held together by a glycoprotein known as J chain. J stands for joining. And it is also synthesized by plasma cells. This J chain binds to the constant region of the IgA monomers through disulfide bonds. So, secreted immunoglobulin A are the IgA antibodies synthesized and released in the circulation by activated B cells or plasma cells. Now let's see what is secretory immunoglobulin A. Secretory IgA is written as sIgA. Now, here S is written in uppercase. These secretory IgA are predominant the mucosal surfaces. It is also a dimer. But while entering the external secretions of the body, it undergoes some modification. Let's understand this further. The B cells, which are located beneath the mucosal surfaces, differentiate into IgA-producing plasma cells. These plasma cells synthesize and release polymeric IgA antibodies. Recall that the J chain is also synthesized inside the plasma cell. So, these are secreted IgA antibodies. These secreted IgA bind to a receptor expressed by epithelial cells of the mucosal surfaces. This receptor is called poly-Ig receptor. This receptor recognizes the J chain of secreted IgA and bind to their C-terminal domains. Once bound, IgA is internalized into a transport vesicle, which moves to the opposite side of the epithelial cell. During the process, the poly-Ig receptor is cleaved enzymatically. The peptide remaining bound to the IgA dimer is called secretory component or piece. This IgA dimer with attached secretory component exits the cell. Now the antibody has become secretory IgA. This secretory piece thus helps the transportation of the IgA into secretions. It also protects IgA from breakdown by proteolytic enzymes found in the secretions. So, now we understand what is secreted and secretory IgA. Let's now talk about the properties of immunoglobulin A. Immunoglobulin A constitutes about 5 to 15% of the antibody concentration in the human serum. It is the major immunoglobulin in the external secretions of our body, such as saliva, mucus, gastric fluid, tears, etc. So, IgA guards or protects the mucosal surfaces of the body. Half-life of IgA is about 6 days. If you remember the previous lecture, we said that immunoglobulin G is the most abundant antibody in the serum. But most abundant antibody in the human body is IgA. If all the production of IgA at mucosal surfaces is taken into account, then it is the most abundant immunoglobulin in the human body in terms of quantity. This is obvious, since humans have about 400 square meters of mucosal surface, which include respiratory, digestive and reproductive tracts. So, the quantity of IgA produced per day in an adult human is more than all other antibodies combined. Let's now talk about the functions of IgA. IgA is the major antibody found in colostrum of milk in nursing mothers. This provides passive immunity to the infant. It protects the infant from gastrointestinal infections during the first few weeks after birth. It neutralizes toxins and pathogens. They prevent pathogens' adherence to mucosal surfaces and thus blocks them from entering the bloodstream. Immunoglobulin A triggers degranulation of granulocytes such as neutrophils. They also play important role in antibody-dependent cell-mediated cytotoxicity, since they bind to the specific Fc receptors on the killer cells. They are poor complement fixers and opsonins.
So students, you just saw the importance of IgA, right? We have seen the process of the formation of a dimer and how it moves to the external secretions, right? And yes, we talked about IgG, that it is most abundant, but that was in case of serum. Okay, so when we just talk about serum, so in that case, IgG is the most abundant. Okay. Okay, so students, any questions, anything related to IgA, any question you have in your mind?
Sir, so in the video, she talked about the two different types of IgA, right? Are those the same as IgA1 and IgA2, or are they different?
Can you repeat the question?
So she talked about sIgA and then capital SIgA.
Yes.
Are those two different from IgA1 and IgA2?
Yes, see, as I said, IgA1 is mostly you'll find in serum, right? And IgA2 is mostly the one that you see in the external secretions. So is that your point?
Yes, sir. So in the video, she had mentioned that the secretory IgA, I think that is mostly found in the serum. And then she said
Yes,
the other IgA is found in, I think, mucosal surfaces.
yes,
Yeah.
yes. And they also showed the difference between the two, right? That one has a longer hinge region. So as I said, when the sequence was carried out, right? So in case of IgA, always two type of sequences were observed. Okay. So as per one of the sequence, you'll get IgA1 and the other one has IgA2, right? So because of this variation of the amino acid sequence, you see a different kind of uh, immunoglobulin, right? So it's basically dependent on the sequence of the constant region domain.
Okay, sir. Thank you.
Yeah. Okay, any other thing that comes to your mind? Okay, so another very important thing is it is present in mother's milk, right? So, because it is there, so the infants, they do not have their own immune system working at that age, right? So, immune system is just developing, it's seeing the antigens and all those things. But at that time, uh, these antibodies present in the mother's milk, so the child is consuming it, right? So that is providing protection to the child in the initial phase, right? And sometime what happens, uh, there are a lot of diseases, right? So the child does not show any symptom till the child is having mother's milk, right? Because the protection is being provided through those antibodies, right? But when the child leaves mother's milk, okay, then they start getting infections, right? So it happens a lot of times. So if the immune system of the child has not developed, okay, so we do not get to know by the time child is on mother's milk, okay. But when the child leaves mother milk, right, then that child becomes susceptible to infections. Okay, so a lot of time you come to know about primary immunodeficiencies at a later stage, right? When this protection from mother's milk is gone. So, no antibodies, the child becomes susceptible to infections, and then when the doctors do the test, then they find out that okay, this specific gene has some mutation, or this particular enzyme is missing, or this particular protein is missing. So you get to know after maybe 6 months or 8 months, whenever the child leaves mother milk. Okay. Okay. So students, any questions? Any other question? Okay, so I'm ending the class and we will meet again after 5 minutes, and in that we will talk about IgE. Okay?